Hello students, myself Popere KS, Assistant Professor in Botany at Red Shikshan Sanstha's RBNB College, Sri Rampur. In this lecture, we are going to study about genetic code. So, first of all, what is genetic code? Genetic code is the set of rules by which information which is encoded in the genetic material that is in the DNA or RNA is translated into the protein. And this is genetic codon. It is a rule by which the genetic information or the code which is present in the genetic material, that is in DNA or RNA, it is translated into the protein. So, and now this encoding is done by ribosome. So this biological encoding means the code which are present in the DNA and RNA, it is detected by the ribosome with the help of one more molecule, that is tRNA. Now this tRNA, it will carry the different amino acid for the synthesis of the polynucleotide chain or the protein. Now this DNA, it is made up of different components, mostly three components, that is a phosphate group, a deoxyribose sugar, and one more group, that is the nitrogenous bases. So this phosphate group and the sugar, that is deoxyribose sugar, it will be similar in all the nucleotide. Only the difference lie in the phosphate group. So only the difference lie in the nitrogenous bases. So this nitrogenous bases, they are ATGC. So only the difference in the DNA, that is sequence in the DNA, will lie in the nitrogenous bases. And suppose if it is RNA, now it will become, so instead of thymine, there will be presence of other nucleotide, that is the uracil will be present. So this is the difference that is between RNA and DNA. Now this DNA, it is made up of it is a chain, or we can say made up of a different nucleotide sequences. Likewise, now this protein, it is also made up of different types of the amino acid. So this protein, it is made up of a different amino acid. Now they are brought about by the tRNA, and with help of the ribosome, they will encode the sequence which is present in the genetic material or DNA and the RNA. So this is the protein and these are the different amino acids. Now the protein, it is made up of different amino acids. Now we will study how this amino acid, they are encoded by the different codon. Now first let us start with the codon. So this codon, it is firstly suppose there are in living world, there are almost 150 different amino acids. So total 150 amino acids are present, but out of that only 20 amino acid, they will code for the different protein. So out of this 150, only 20, that is the 20 will be coding for the different protein. Now how this different types of amino acid, they are means how what are the code for this different 20 amino acid. So now the sequence of the nucleotide that code for one amino acid is called as a code. First, suppose we think that the codon is a singlet, or we say single codon. Single codon means there are 4 nitrogenous bases. So this is 4, and if we means suppose a nucleotide base, that is one base will code for one amino acid, so what will happen? Only 4 proteins can be coded, or 4 different amino acids can be coded from this if the codon is singlet. And if it is a double codon, so what will happen? It will code only for the 16 different types of the amino acid. As there are 20 amino acids, so this is not enough for the amino acid which is coding for the protein. Now next is triplet codon. So if we consider that the codon, that is the 4 nitrogenous bases, we take them in the triplet form. Now this will code for the 64 different types of amino acids. Now the number 64, it is enough for the synthesis of the, that is for the 20 different amino acids for the protein synthesis. Now so this 64 codon, this were considered means for the synthesis of the protein, that there are different 64 types of the codon which will code for the protein synthesis process. Now we will see the genetic codon, the triplet form of the genetic codon. Now this is the genetic codon. Now here there is a it can be found out by making a table. It will have a 16 different column. Now from this side U, C, A, G will be taken, and from upper side also U, C, A, G will be taken, and for this first there will be written U, C, A, G. Now how this coding will be done? First you have to take U from this side, next the U from the upper side, and this U from this side, that is, now it will become U, U, U. Now next again U, U and C from that side. Likewise UUA, UUG. Now this UUU and UUC, it will code for a amino acid, that is the phenylalanine. Now this UUA and UUG, it will code for the amino acid leucine. Likewise we can find out the different codon which will code for the different types of, that is for the 20 different types of the amino acid. Likewise this CUU, CUC, CUA and CUG, it will code for the amino acid leucine. Now this AUU, AUC, AUA, it will code for the amino acid isoleucine. Now this next codon AUG, this is the codon which will code for, now it will code for methionine, and also it will be acting as a start codon. So for during the translation process, that is the protein synthesis process, what will happen? This codon will be incorporated first by the tRNA, that is the AUG, so it is also called as the start codon. Likewise there are different codon which will code for the different amino acid. The next, this is GUU, GUC, this will code for valine. Likewise there is different codon for serine, proline, threonine, alanine. Now this will code for the tryptophan. Now there are different codon like histidine, glutamine, aspartic acid, lysine, aspartic acid, glutamic acid, arginine, serine. There are so these are the different types of the codon which code for the different amino acid. Now one thing is there, that is UAA, UAG and UGA. These 3 codon will not code for any amino acid. These are acting as a stop codon. Stop codon means when this codon will appear on the mRNA during the protein synthesis process, what will happen? The translation process will terminate there, as this codon has appeared on the mRNA. So these is the different types of the codon which will be code for the amino acid. Now we will see the properties of this genetic codon. Now first is, so as we have already seen that the genetic codon, it is a triplet. So the codon will be in form of a triplet. So suppose like first only UUU, it will code for the amino acid that is the phenylalanine. So first property is that the genetic codon is triplet, because if we take single or double, it will be not enough for the coding of the 20 different types of the proteins. Now next is universal. That is, this code or the codon will be universal, that is from the lower organism, or we can say from amoeba to the largest organism, which will be similar for all the organisms. Suppose this UUU, it code for the phenylalanine, and GUG, it will code for valine. So it will be similar in case of this any smaller organism, or we can say from amoeba to the higher organism. This two will only code for phenylalanine, and this codon will code for valine only. So the genetic codon, it is universal. Now third is commaless. So what does it mean? Commaless means, so when this codon, they are present on the mRNA, there is no spacing between this codon. So suppose this is a mRNA, it will have different codon. So this codon, the triplet codon, they are in continuous form. There is no spacing or space between this triplet codon. So this is a property, that is the genetic codon, it is a commaless. Now next property is ambiguity. Ambiguity means the uncertainty. So the genetic codon, it is non-ambiguous. So this genetic codon, it is non-ambiguous. So this UUU, it will code for phenylalanine in all the organism. So it will happen in vivo, that is in vivo condition, so in the natural condition. But suppose if we kept kept the streptomycin, that is in, so what will happen in presence of the streptomycin, what will happen, this UUU, it will act for the different amino acid, that is valine, lysine or serine. So this genetic codon, it will be non-ambiguous in natural condition, but in artificial condition or in vitro condition, on presence of certain chemical, what will happen, this will code for the any other different amino acid. So now we can say that this codon is ambiguous, but in natural condition the genetic codon is non-ambiguous. Now next is genetic codon is degenerate. So this degenerate means there are total 64 codon, and out of which 61 code for the different amino acid. As the 3 codon, the rest of the 3 codon, they are acting as a stop codon. Now this amino acid degenerate means the one codon will code for a specific amino acid. So degeneracy means when amino acid is coded by more than one codon, such property is called as degeneracy. So suppose this UUU is a codon for the phenylalanine, and one more, that is UUC, is also a this codon also code for the phenylalanine. So this phenylalanine, it is coded by the two different codons. So this is called as degeneracy. Degeneracy means the one amino acid is coded by the different, means more than one codon. So this is called as degeneracy. Now one more property is there, that is the genetic codon. It is non-overlapping. So non-overlapping means that there will be the 3 codon will be coding for a separate amino acid. That 3 codon will code for only one amino acid. So suppose these are the 6 sequence. Now this will code for one amino acid and this will code for the next amino acid, means there is no overlapping of the codon. Now overlapping means this first will code for one amino acid, now this next will code for the second, like this will code for the third amino acid, and the last one, this will code for the fourth amino acid. So if the codon is overlapping, there is formation of 4 different types of the protein. But as the codon is non-overlapping, from this 6 bases there is formation of only 2 different types of the amino acid. So these are about the properties of the genetic codon and about all the genetic codon. So genetic codon, it is a triplet, and one triplet codon will code for one amino acid for the synthesis of the protein or the translation process. Thank you.